Hello everyone, welcome back to Teacher Kayal's Guide to Biology A+. In this video, I'll guide you through answering SPM trial questions on the topic Chemical Compositions in a Cell, focusing on nucleic acid and water. There are two types of nucleic acid, which are DNA and RNA. Both of them are made up of nucleotide monomers. Each nucleotide consists of a phosphate group, a pentose sugar, and a nitrogenous base. DNA consists of two polynucleotide chains forming a double helix, while RNA consists of only one polynucleotide chain. The polynucleotide chain in DNA is longer, while the polynucleotide chain in RNA is shorter. In terms of nitrogenous bases, DNA contains thymine, adenine, guanine, and cytosine, while RNA contains uracil, adenine, guanine, and cytosine. As I previously mentioned, DNA consists of two polynucleotide chains forming a double helix. Gene mutation causes a change in the genetic code, so the codon in the mRNA will also be different. Consequently, a wrong amino acid will be added to the chain, forming an abnormal protein that is usually useless. DNA is a macromolecule, meaning it is a very large molecule made up of many repeating nucleotide units. Each nucleotide consists of a phosphate sugar, a pentose sugar, and a nitrogenous base. DNA is made up of two polynucleotide chains that twist to form a double helix structure. The two polynucleotide chains are held together by either two hydrogen bonds or three hydrogen bonds between the nitrogenous bases. The four nitrogenous bases in an RNA molecule are uracil, adenine, guanine, and cytosine. The diagram shows a part of a DNA chain. To complete the missing nitrogenous bases, we need to follow the complementary base pairing rule. Thymine pairs with adenine, cytosine pairs with guanine, and adenine pairs with thymine. When you are required to draw and label a nucleotide, you must read the question carefully to check whether you need to draw general nucleotide, DNA nucleotide, or RNA nucleotide. You must draw a circle to represent phosphate group and draw pentagon to represent sugar. But you can draw any shape to represent nitrogenous base. For the phosphate group and nitrogenous base, label them the same regardless of the type of nucleotide. However, for the sugar, the label depends on the question. If the type of nucleotide is not specified, label the sugar as a pentose sugar. If specified as DNA nucleotide, label the sugar as deoxyribose sugar. If specified as RNA nucleotide, label the sugar as ribose sugar. For this question, you are required to draw a polynucleotide chain made up of three nucleotides. Ensure that the phosphate group and nitrogenous base are attached to the correct vertices of the pentagon representing the sugar. When you are required to complete the drawing of a polynucleotide molecule, draw the polynucleotide chain in opposite directions. Ensure the pentose sugar is also drawn in opposite orientations. The nitrogenous bases must have complementary shapes, with 2 or 3 hydrogen bonds linking each pair of nitrogenous bases, because adenine pairs with thymine via 2 hydrogen bonds and guanine pairs with cytosine via 3 hydrogen bonds. The nucleus contains most of the cell's DNA, while mitochondria and chloroplasts contain only their own DNA. The DNA contains genetic code. The genetic code carries hereditary information. This determines the characteristics of each living organism. DNA is the master copy of genetic instructions. In protein synthesis, first the genetic instructions are transcribed to form mRNA. The mRNA is then translated into a sequence of amino acids, forming a polypeptide chain. The polypeptide chain then folds to form a protein. If transcription does not occur, mRNA cannot be formed. Amino acids cannot be arranged into a polypeptide chain, so no protein can be synthesized. If DNA is degraded, genetic information cannot be inherited, so the characteristics of living organisms cannot be determined. In addition, degraded DNA cannot be transcribed to form RNA. One important property of water is high specific heat capacity, which means water can absorb a large amount of heat energy with only a small increase in temperature. Therefore, water has to maintain a stable temperature for metabolic processes. So, when we drink a lot of water during fever, the water can absorb a large amount of heat energy without a big increase in temperature of the water. In this video, I've covered most of the 2021 to 2025 trial questions for this part of the syllabus. If you like to have the complete set, feel free to contact me through the email address stated in the description. Remember, study smarter, not harder.